In the previous lecture, I told you how cells use energy stored in the phosphate bonds of ATP to make otherwise endergonic anabolic reactions run uphill by donating energy to those reactions. Remember that the basic way this is done involves a two-step process. First, some of the energy stored in ATP is transferred to one of the substrates of the reaction, a process that's exergonic and thus is likely to run spontaneously. Then, once the substrate has this added energy, the substrate is phosphorylated, as we say, a reaction that was endergonic becomes now effectively exergonic because of the higher energy state of the phosphorylated substrate, and this reaction will now run spontaneously. The ability to couple the energy released from exergonic reactions to power endergonic reactions is critical to making the chemistry of life work, because it makes it possible for cells to run energy-requiring anabolic reactions. And this is important, don't forget. The synthesis of the complex biomolecules that make up living organisms all are anabolic reactions. But we still have another major issue to address in order to understand how the chemistry of living things works. Just figuring out how to make endergonic reactions run energetically uphill isn't enough. We also have to figure out how cells control these biochemical reactions, how they control both exergonic and endergonic reactions. Obviously, not every reaction that could run in a cell should run all the time. And when reactions that should run do run, the cell needs to make sure that they run at appropriate speeds. Now, as it turns out, most biochemical reactions in cells, even exergonic reactions, don't run spontaneously ever. Instead, they need some assistance to get going. In today's lecture, we're going to look at why this is, and we'll also look at how biochemical reactions are controlled in general. Now, to do so, we're going to go back and look in more detail at some proteins that are by now familiar friends. And of course, the proteins I'm referring to here are those that function as enzymes. Let's begin this discussion by considering once again the sugar glucose. In the last lecture, I used glucose as an example of a complex, energy-rich molecule, contrasting it to a collection of carbon dioxide and water molecules that are made of exactly the same kinds of atoms in the same numbers, but are arranged a little bit differently, and that have relatively little energy stored in them. Remember also from last time that more orderly high energy states are generally less stable because they have a lot of free energy, and that as a consequence of this fact, these more orderly high energy states, like glucose molecules, might spontaneously change their state to something that's less orderly with lower energy. So with this background, you might think that glucose should spontaneously break down into carbon dioxide and water, but it usually doesn't do so. In fact, most exergonic reactions in the cell, that is, reactions that will release energy and should run spontaneously because they run downhill energetically speaking, don't actually ever run spontaneously. Why not? Well, the reason for this is that even the exergonic reactions that require, or even, the reason for this is that even exergonic reactions usually require the input of a little bit of energy to start breaking the chemical bonds of that high energy substrate. Now you see, most chemical bonds are inherently stable, even high energy ones, at least to some degree. Some bonds are more stable than others, but in general, the chemical bonds that are formed between atoms in a molecule tend to resist being broken. For this reason, even high energy molecules have to have their bonds destabilized a little bit by adding even more energy to the molecule before those bonds will begin to break, allowing the atoms in the molecule to rearrange themselves into some other, more stable, energetically lower configuration. The energy that needs to be added this way to get an exergonic reaction to run is referred to as the activation energy of the reaction. Now, to get a better idea of what activation energy is, let's picture again, as we did in the previous lecture, a graph that has time on the x-axis and the relative amount of free energy of compounds on the y-axis. Now, last time, we illustrated an exergonic reaction by drawing a straight line from a high-energy reactant to a low-energy product, a straight line going down. Or I should say, a line that is moving straight down at an angle. However, a more realistic drawing for the energy relationships of a typical exergonic reaction in the cell would not be a straight downward line, but would instead be a curve that initially went upwards to a higher energy level and only then went downwards, down to the lower energy state of the reaction's product. The product of the reaction is at a lower energy state than the reactant at the beginning, but there's sort of a hill that has to be crossed to get from the high energy state to the lower energy state. You have to go a little bit higher in energy. In fact, the image of a hill fits well if we go back to our example of a ball rolling down a ramp. If the ball is at the top of the ramp and there's no barrier, it will very likely just roll down the ramp. However, imagine that the ball is sitting in a depression at the top of the ramp. It won't roll down because it has to get over the little bump on the side of the depression before it gets to roll down that ramp. It needs some energy to get itself out of that depression, rolling it uphill a little bit before it then can roll spontaneously downhill. This added energy that's needed to get the ball rolling, so to speak, is what we call the activation energy of the reaction. Once the activation energy has been added, then the reaction can proceed downhill. This is still an exergonic reaction, even though we have to put some energy in at the beginning, because energy is released overall. That is, the product is at a lower energy state than the reactant. It's just that we needed to add some energy at the beginning to get things started. Now, you might be thinking this seems like an unnecessary complication that we have to deal with in order to understand how the chemistry of cells works. After all, we have enough to keep track of here. We have to keep track of the difference between anabolic and catabolic reactions, figuring out how to deliver energy needed to get anabolic reactions to run uphill spontaneously, and so forth. However, the fact that even exergonic reactions in the cell require an activation energy before they'll run is really a good thing. In fact, it's essential for life to exist. Remember that most of the molecules in our bodies are high-energy complex molecules, including things like proteins, nucleic acids, carbohydrates, and fats. If these high-energy molecules spontaneously broke down at normal temperatures, then literally we'd spontaneously combust. In fact, to be more accurate, we'd never get put together in the first place. Living things are made out of these high-energy complex molecules, and these complex molecules can only exist without spontaneously breaking down because they require an activation energy for that breakdown to occur. Okay, so now we've learned that even though high-energy molecules are less stable than low-energy ones, and even though exergonic reactions that convert high-energy reactants into low-energy products are energetically favorable and will run downhill, we know now that they usually don't do so spontaneously because the high-energy reactant needs to be pushed up a little bit to an even higher energy state in order to begin to destabilize its chemical bonds. And this explains why the complex molecules that make up cells don't simply spontaneously break down all the time. At least not at a very fast rate. Now, as an aside, I should point out that actually exergonic reactions will eventually run spontaneously. It's really a matter of rate. Given enough time, a high-energy molecule of glucose might well break down. It's just that it's going to take a long time to do that. You might think back to our analogy of a ball at the top of a ramp caught in a little depression. It's as though the ball is constantly being jiggled a little bit, just a little bit. And every once in a while, very rarely, that jiggling might be just enough to get it over the bump and have it run downhill. Now, if we add energy and jiggle it even more, actually push the ball up, then it will run down. But spontaneously, the ball might run down every once in a while, just like glucose might break down every once in a while. But without activation energy being added to these reactions, this breakdown will only occur at what's really effectively an infinitesimally slow rate. The importance of activation energy for allowing reactions to run suggests one way we can control whether or not a reaction will run. Specifically, when we do want a reaction to run, we simply have to add the necessary activation energy, right? This is what chemists do when they cause reactions to run in the lab. Unfortunately, the approach that chemists take won't work very well in living cells. Let me explain why not. If we're chemists and we want a reaction in a test tube to run, we generally provide the necessary activation energy in the form of heat. For example, we might take that test tube containing the reactants of the product that we're thinking about and place it over a Bunsen burner. heating the solution just enough so that the heat energy raises the energy level of those reactants and then causes the reaction to overcome the activation energy and then go downhill spontaneously. This approach works well for chemists in the lab, but not so well for living cells, for at least a couple of reasons. The first problem is that the kinds of temperatures needed to provide the activation energy of many catabolic reactions in cells are so high that those temperatures would kill the cell. Don't forget that if you heat up proteins, for example, they fall apart. They do what we call denature. Now, even if the high temperatures weren't a problem for the cell, we'd still be left with the fact that we can't control reactions in a selective fashion this way. There's no way to significantly change the temperature of just one part of the cell and not others. So the cell can't add activation energy selectively to some reactions but not others simply by heating things up. We need a more controlled way to do this. Fortunately, there is an alternative way to get reactions to run faster. Instead of adding a lot of activation energy, this alternative is to lower the amount of activation energy that's required. Now, if we think back to our graph of energy relationships in a reaction, this approach would be seen as a lowering of the height of the activation energy hill that must be surmounted. So how can we lower the activation energy of a particular reaction? Well, in general, this is done by substances that we refer to as catalysts. Catalysts are substances that increase the rates of chemical reactions by lowering the activation energy needed for that reaction to occur. Catalysts interact with the compounds involved in a chemical reaction, that is, the reactants, but they're not themselves reactants. They just interact with the reactants and stay out of it otherwise. Now, there are many non-biological catalysts that are used by chemists in research and industry to make reactions run faster. For example, powdered platinum can be used to catalyze any reaction that involves hydrogen as one of the reactants. This is because molecular hydrogen, that's H2, interacts with platinum in such a way as to destabilize the bond that holds the molecule together. Thus, catalysts overcome the problem of needing to add a lot of activation energy because they reduce the amount of activation energy needed in the first place, and they usually do this by somehow interacting with one of the substrates of the reaction of interest. However, most non-biological catalysts, such as platinum, are very highly nonspecific. That is, they'll catalyze a wide range of chemical reactions. Any reaction involving molecular hydrogen, for example, will be catalyzed by platinum. In cellular metabolism, in the biochemistry of cells, it's important to be much more selective in the reactions that are catalyzed. This is where enzymes play their essential role in making life's chemistry work. Enzymes are those remarkable proteins that solve the dual problem of lowering the activation energy necessary for a metabolic reaction to run and of doing so in a highly selective fashion. We've heard a lot about enzymes throughout this course. Most of the molecular processes we've talked about have involved enzymes in one way or another to make them work. Sometimes a small army of enzymes are involved. Let's take another look at enzymes now, specifically from the point of view of how they facilitate biochemical reactions in the cell. Enzymes are biological catalysts. An enzyme speeds the rate at which a reaction occurs by lowering the activation energy barrier to a more modest level, a level that's within reach even at the normal moderate temperature of a living organism. Like any catalyst, enzymes don't actually change the overall energetic relationship of a reaction. That is, the enzyme doesn't change the difference in free energy between the reactants and the products of a reaction. Or to put it another way, enzymes can't by themselves make endergonic, that is, energy-requiring reactions run uphill. All that enzymes can do is to lower the activation energy. Now, once that activation energy is lowered, once the hill that has to be overcome energetically is set to a much lower level, then the rate at which the exergonic reaction will occur spontaneously increases dramatically. This change can be astronomical, effectively serving to turn a reaction on or off. Without the enzyme, the reaction will run so slowly it might as well not run at all. With the enzyme, the reaction runs at some reasonable pace for the cell. Here's a typical textbook example to make this point. Consider a protein that, you remember, that's just a string of amino acids that happens to have the amino acid arginine as the last amino acid in the polypeptide chain. The peptide bond, that's the bond that holds this terminal arginine to the rest of the polypeptide. The peptide bond is a relatively stable chemical bond, but it has a high energy, and therefore, given enough time, this bond might spontaneously break down. Now, it's got to take a lot of time. For example, one estimate is that the half-life of this bond, that is the time it takes for, if you took a population of these proteins and waited until half of them lost an arginine spontaneously, the half-life of this bond is about 7 years. Start out with some number of these proteins. 7 years later, half of them will spontaneously have lost one of the arginine at the end. But there's an enzyme called carboxypeptidase A. And this enzyme actually specifically catalyzes the reaction that cleaves a terminal arginine from the rest of a polypeptide. Now, if we add this enzyme to whatever container we have these proteins in, then the half-life of the peptide bond that's holding arginine in place decreases from 7 years to half a second. In other words, the amount of reaction that would have taken place in 7 years spontaneously is reduced now to just half a second, because this protein, acting as an enzyme, has lowered the activation energy required to make that particular reaction run, the reaction that splits off the arginine. Now, unlike non-biological catalysts, such as the platinum example we gave, enzymes are very, very selective in the reactions they catalyze. That is, each enzyme is specialized for catalyzing only a single reaction. We've alluded to this point many times earlier in the course, in the context of discussing how the structure of a particular protein determines its function. Let's now ask ourselves how this actually works in a little more detail. Enzymes have sites that will bind one or more of the reactants of whatever reaction they catalyze. Now, we call a reactant that binds to an enzyme the substrate of that enzyme. And we call the place on the enzyme, the physical place on the protein where that substrate binds, we call that the active site of the enzyme. The specificity with which an enzyme binds a particular substrate depends on the exact three-dimensional shape of the active site and on the particular physical and chemical characteristics of the amino acids that make up that binding site. And only a very narrow range of substrates, sometimes only one single kind of molecule, can actually fit into the active site of an enzyme. This kind of specificity, depending on the shape of a protein, is nothing new to us. It's just another example, actually perhaps the premier example, of how structure determines function in proteins, a point we've kept coming back to repeatedly. Now, the way that enzymes interact with their substrates, therefore, is often analogized to the physical relationship between a lock and a key. You might see the active site of the enzyme as being a lock and the substrate fitting into it in a very specific way, just like a key would fit into only one lock. And in fact, the earliest descriptions of how enzymes worked emphasize this lock and key relationship. As it turns out, however, the physical structure of the active site of an enzyme is not necessarily rigid in the same way we think of a lock being rigid with respect to the key that fits into it. In fact, in many cases, the interaction of a substrate with the active site of an enzyme causes that active site to change its shape itself. In other words, as the substrate enters the active site, the enzyme will change its shape slightly, causing that substrate to fit even more snugly into position. This would be as though a lock changed its shape to envelop even more of the key as the key was first being inserted into it. Another image that's often used to describe this process is that it's as though the enzyme clasps its substrate, the way you might clasp another person's hand when you're shaking it. Now, we refer to this more dynamic view of the physical interaction between an enzyme and its substrate as the induced fit model of enzyme function. Induced fit because the fit of this enzyme is induced to become even closer as a result of binding with its substrate. And we contrast this induced fit model with the older lock and key model. This induced fit the change in the enzyme shape, serves to bring whatever chemical groups that are associated with the active site of the enzyme into even closer, more intimate proximity to the substrate in a way that will help catalyze the reaction. Now, once a substrate is bound to the active site of an enzyme, the active site does catalyze the conversion of this substrate into whatever product is formed by the reaction, and the product then drops off the enzyme. This same enzyme molecule then will then interact with another substrate molecule, and the catalytic cycle, as it's called, repeats. In other words, the enzyme isn't used up in this reaction. It interacts with substrates, it does stuff to them, the substrates become products, they drop off, and the enzyme then starts over and works on some other substrates. The turnaround time for an enzyme to act on a single substrate, in other words, to catalyze one reaction, is usually very, very fast. A typical enzyme, for example, might go through 1,000 substrate molecules a second. And in fact, some enzymes are much faster than this. Given this rapid turnaround, and given the fact that enzymes aren't used up by the reactions they catalyze, it only takes a very small amount of a particular enzyme in a cell to have a major impact on the overall rate at which a metabolic reaction will occur. Well, the question we really haven't answered yet, though, is how do enzymes actually do their work? In other words, how do enzymes actually lower the activation energy? What are they doing to their substrates? Well, what they do is a variety of things. The way they lower activation energy occurs in a variety of different ways. Some are quite simple, and others are more complex. One of the most simple and fundamental ways in which enzymes catalyze reactions is by bringing two or more substrate molecules together in close proximity and in the proper orientation relative to each other. We saw an example of this when we looked at the function of amino acid tRNA synthetase. Remember, that enzyme has two binding sites, one for a particular amino acid and one for a particular tRNA that corresponds to that amino acid. These binding sites are actually in close proximity on the enzyme, and what they're doing is bringing the amino acid and the tRNA just in close proximity to each other, bringing them together in the right orientation to facilitate the building of a bond between them. That's why we referred to this molecule as a sort of molecular matchmaker. Now, another common way for enzymes to catalyze reactions, to lower the activation energy, is to impose some sort of molecular stress on the substrate. As the substrate and the active site fit together, not only does the enzyme change its shape, as suggested by the induced fit model, but also the active site of the enzyme may affect the shape of the substrate as well. This shape change literally puts a kind of molecular tension on some of the bonds in the substrate molecule, stretching them out, bending them a little bit, and therefore making them less stable and allowing them to be broken more easily. If the activation energy involves the energy needed to start destabilizing bonds, what the enzyme is doing in this case is actually destabilizing those bonds by bending the substrate molecule. Finally, the amino acids that are present around the active site of an enzyme might actually interact chemically with the substrate. For example, by adding hydrogen atoms or other ions and chemical groups. And this chemical exchange also can destabilize the substrate molecule, again making its chemical bonds more easily broken and thus lowering the effective activation energy. Well, we've looked a little bit at how enzymes do their work, but there is still yet one more basic issue we need to address to understand how the chemistry of life works in cells. The question is this: what regulates the activity of enzymes themselves? We know that enzymes can work on these reactions, but what makes these enzymes either do this or not? Remember, the goal here is to have reactions running when they want them and not running when we don't want them. Well, we already know at least one part of the answer to this question. We've seen before how not all genes are expressed all the time in a cell, but instead that the expression of genes is highly regulated. Thus, one fundamental way in which a cell can regulate enzyme activity is by only expressing the genes that are coding for those particular enzymes at times when the metabolic reactions catalyzed by those enzymes should be active. We saw an example of this when we talked about gene regulation of the lac operon in prokaryotes. Remember, the enzymes beta-galactosidase and galactosidase permease are necessary to break down lactose, for lactose to be digested, but these genes are only allowed to be transcribed when lactose is present. So the genes themselves are turned on and off. Now, we made a point, though, when we talked about gene regulation, that this approach is a relatively slow way to regulate the activity of proteins in general. Often it's more adaptive to have proteins on hand already so that they can be used immediately when they're needed. There are two other ways, other than gene regulation, that a cell uses to control the activity of enzymes that are already present in the cell. Now, to understand these two mechanisms, let's first go back to a point I touched on at the beginning of the previous lecture, which was to note that the metabolic reactions in a cell are usually interconnected as a series of metabolic pathways, with the product of one step serving as the substrate for the next step. For example, the amino acid threonine can be converted into another kind of amino acid called isoleucine. And this conversion is done commonly, but it involves a series of 5 steps, with each step catalyzed by a different enzyme. In other words, there are 4 intermediate products in the metabolic pathway that leads from threonine to isoleucine. The first enzyme converts threonine to the first intermediate, then another enzyme comes in and converts that intermediate molecule to yet another intermediate molecule, and we have to go 5 steps down the line until we get the desired end product, which is isoleucine. In metabolic pathways such as this, it's often the case that one step, the first step, serves to regulate the rest of the pathway. If threonine isn't converted into the first intermediate product, then the rest of the pathway comes grinding to a halt because it has nothing to work on. The substrates are not produced that get sent down the line. But how can cells regulate the activity of one step in a metabolic pathway? Well, one way to do this is to separate the enzymes and substrates into different compartments of the cell. Remember, eukaryotic cells are not just a bag of chemicals, but instead are very highly compartmentalized structures. Thus, a way that a cell can regulate metabolic reactions is to separate particular enzymes from their substrates, keeping them apart in separate chambers across membrane barriers, and bringing them together only when that metabolic reaction should run. Now, there is a disadvantage to this approach, which is largely that it requires some mechanism to transport substrates across cell membranes. And this is done, but it often also requires the addition of some energy to do so. And it also doesn't provide a very fine-tuned way of controlling metabolic processes. Probably the most important way in which cells regulate metabolic activity is through what's called feedback inhibition. Feedback inhibition occurs when a metabolic pathway is switched off by its own end product. For example, in the pathway that leads from threonine to isoleucine, the end product, isoleucine itself, will bind to the first enzyme in the chain of reactions. Now, this binding of the end product to an enzyme that's somewhere mediating a reaction along the pathway actually usually occurs at a different site on the enzyme, different from the enzyme's active site. But as we've seen many times before, when this end product, isoleucine, binds to this intermediate enzyme, what happens is that the enzyme will, as a result, change its shape. It changes the shape from an active form to an inactive form. So the binding of isoleucine itself essentially turns off one of the enzymes in the metabolic pathway, preventing that metabolic pathway from running. Essentially, the end product feeds back and renders one of the critical regulatory steps dysfunctional. How does this feedback actually act in regulation? Well, for example, as isoleucine accumulates, we may not need to make more isoleucine. We've got enough. And so it makes sense that this end product, the isoleucine itself, should feed back and turn off what's essentially the production line for making more isoleucine. Now, this is a simple example, and the ways in which products interact with enzymes in metabolic pathways can be much more complicated and subtle than this. Nonetheless, this example illustrates how the interacting pathways of metabolism, with feedback inhibition and other ways in which the activity of enzymes can be increased or decreased relative to other enzymes, how this provides a sophisticated web of control, allowing cells to fine-tune their metabolic activities as the needs of the cell change. We've begun our exploration of energy in living systems by looking at some of the fundamental issues involved in making life's chemistry work. Our next step will be to look at the processes involved with cellular respiration to see how living things actually use the breakdown of sugars and other carbon compounds to manufacture ATP, which is then used as the common currency of the cell. And we'll begin this story in the next lecture.